take it away.
Thank you. Thank you, Gerd, and Guido and Isabella. It's great to be here. Well, I'm home, but great to be almost there. And I'll share my screen. Sorry, I must have closed the PowerPoint. Okay. So today we'll be talking about inherency and agency in the evolution of development. And I'll start with a big question. So how did we get from here, the first cells almost 4 billion years ago, to the diversification of life, this ramified set of organisms that kind of find their own way in the world, have this whole variety of different structures. You can see the different landmarks along the way. First eukaryotic cells greater than 1.5 billion years ago, the first animals around 700 million years ago or more, the complex body plans of the Avalon explosion, the Cambrian explosion, and then the plants about 500 million years ago. How did this all happen? So the default view for many years was a kind of a gradual set of changes where you would have incremental variations and selection for increased fitness and adaptation. And eventually through these microevolutionary processes, you would get these large scale changes and varied diversification. But we know now that these things didn't happen so gradually. There were fits and starts and major transitions, and some of them happened fairly abruptly. And we know that a big part of that is the role of development in creating non-random variations. But how did development do that, and how did developmental mechanisms themselves evolve? So let's just look at this tree of life and just naming the different types of organisms. Now, these are all of the organisms that we know and the relations in a very schematic way. And some of these forms have multicellular counterparts, and some of them don't. And if we look here, there are multicellular bacteria, there are multicellular amoebozoa like Dictyostelium, Dysploidium, multicellular plants and algae, and then of course the group that we belong to, the opisthecons, which include the animals and the fungi and single-celled organisms or quasi-multicellular organisms that are very closely related genetically to the animals or metazoa. So as you'll see, I'm going to focus most on this group, but I'm going to make the proposal that much of multicellular evolution, rather than resulting from cycles of gradual selection for adaptive advantage, was driven by inherent physical properties of multicellular matter and the optional recruitment of ancestral unicellular functions to specialized cell and tissue types. This will become clearer as I go on, but function is a big question in evolutionary biology and in the philosophy of biology, and where do functions come from? So I'm going to first talk about where forms came from, and in my view, and then where did functions come from? Are they simply a matter of random search, or is there something inherent about biological function? And then I'm going to be talking about the agency. Even individual cells seem to have motives and goals and basically things they want to do. So there's a kind of a residual factor, which is possibly one of the most important in the evolution of animals. Where does agency come from? I'm going to be talking about the progressive alignment of ancestral single-cell agency to the emerging motives of multicellular entities. Okay, so all of this verbiage will become clear as I give examples. So let's start with the inherency of form. Well, then we'll go on to this and this. So multicellular organisms are materials, and they are different kinds of materials, and those different kinds of materials have different inherent motifs and properties. So for example, let's take a simple example. If we look at cells that are related to the animals, they are sometimes multicellular, but they are somewhat inert. The cells are embedded in stiff matrices, and you have some multicellular forms, but there's not a big variety among the forms that you get. The cells can't really do much when they're embedded in these stiff matrices. We have multicellular amoeba, cellular slime molds, for example. And here you have cells that themselves are motile, and they're embedded at different points in their life cycle in a kind of a very compliant, viscous, soft matrix, and they can form complex structures. But they can't form structures with internal cavities because the materials that the cells are embedded in are kind of soft, and they kind of, any spaces close up, and they can do some things, but not a lot of things. Then we have organisms like ourselves, the animals, the metazoans, and these cells are connected to each other in a transient but strong fashion. So the cells remain connected and they can move past each other. And this gives them the properties of a liquid. They're kind of a viscous liquid, and liquids can have many different structures, and you can have liquid crystals, and you can have polar liquids that can have internal spaces. So there's a lot of kind of inherent forms that can arise from this kind of material. And because we know a lot about the physics of liquids, we can say that some of these forms resemble non-living liquids, and we know a lot about the physics, so we can predict what the forms might be. So this is an interesting thing. Just because of the kind of material, we have a kind of a predictability about the forms. Plants are a different story entirely. The cells are embedded in stiff cell walls, a matrix, but the cells are connected to each other through plasmodesmata, these connecting channels. And this is a very active tissue, and the cells can expand and contract, and they can produce enzymes that melt the matrix. This is a material that is very dynamic, and you can get many, many structures and forms in plant tissues, but it's not a physics that we're that familiar with. It's not the kind of generic physics of liquids. So I'm not going to be talking about plants. I've written about it with colleagues, Mariana Benitez and Carl Nicholas, but this is a very exotic form of material. I'm going to focus on the liquid-like materials of the animal tissues. Okay, so let's look here. We're still in the opisthecons, but we're focusing on the metazoans, the choanoflagellates, and the ichthyospores. They're all genetically related, but they have different characteristic motifs. Okay, and then the metazoans I'll focus most on. Okay, so the organisms in the blue circle, the metazoans and the choanoflagellates, the ichthyosporians are called holozoans. And the holozoans have genes that specify things that were eventually used in animal development. But of course, they weren't used in development in single-celled organisms. They were just there to do different things that they evolved to do in single cells. But when the cells became joined together through a set of proteins called cadherins that made them this liquid material, then these genes and gene products took on new functions in the multicellular context. Somehow the physics that's mobilized in the multicellular material is quite different from anything that goes on in single cells. And then new genes were also acquired after you got the first animal. So cadherins cause clustering, and a special kind of cadherins that link up to the cytoskeleton called classical cadherins allow these clusters to become very cohesive, despite the fact that the cells can move with respect to each other. And you can get epithelioid tissues, tissues where the cells are very closely connected, but the cells can move. And you can get polarity of the cells, so that gives you interior spaces. I'll get into that. But then you acquired new genes after the origin of the animals that allowed for the cells to become what's called planar polarized, and that allowed them to orient themselves. The tissues, instead of just rounding up, could then elongate by a process called convergent extension. And you also got an enzyme called peroxidase that was not present in these earlier holozoan forms that polymerized collagen. And when that happened, you got these planar substrata called basal lamina. And the basal lamina allowed the cells to spread and remain in separate sheets. And you could get very complex structures when you take the toolkit that came along with the early holozoans and added to them some of these extra genes and gene products. And then you could get multi-layered structures. You could get cells coming apart and disaggregating within the context of the whole organism. So you can get a complex morphogenesis. And so I'm just going to use an analogy here. So non-living materials exhibit characteristic morphological motifs that depend on their composition and the mesoscale or middle scale physical processes mobilized by the materials. So we're used to water under different circumstances or other liquids having different forms that are characteristic, vortices and waves and so on. Solids can form crystals, and depending on what the solids are made of, they can form different kinds of structures. So we're familiar with the idea that, that materials have characteristic motifs, that you don't really have to search very hard when you have these materials to get these forms that are recurrent again and again. With certain variations, they always form the same kinds of structures. So similarly, the liquid tissues of metazoan organisms have characteristic motifs because the molecules within them that they secrete and communicate with each other by also mobilize physical forces, specific physical forces and effects that cause these multicellular materials of the metazoans to form particular structures. So I'm sure many people are familiar with the idea of gene regulatory networks. And we know that genes and their products interact with each other and cause switching between different states of cells. But dynamical patterning modules are beyond that. They require gene regulatory networks, but they also require physical forces and effects that allow multicellular structures to form patterns and morphologies. You can get segmentation, kind of buckling and multilayering, and kind of salt and pepper configurations of different cell types. All of them involve cell differentiation, but there is a physics that organizes the differentiated cells, and that physics is mobilized by the particular proteins that are characteristic of different stages of metazoan evolution. So let's look at this chart. So these are dynamical patterning modules, as I've defined them. They just have acronyms, and there are characteristic molecules that mediate these effects. So like you have these physical forces beyond the molecules, the force of adhesion, lateral inhibition, where cells are instructing cells next to them not to do what they're doing, to do something else. You have differential adhesion, cell surface anisotropy, cell shape anisotropy, and so on. And this gives you the different characteristic motifs. Just the way liquid water can have waves and can have vortices, the liquid materials that make up metazoan organisms have characteristic motifs that are defined not only by the molecules, but by the physics that is mobilized by the, and then you can get these different forms of patterns. So you can see that this is the first kind of indication of the predictability that you don't really have to search very far to utilize cells and these toolkit genes and their products to produce characteristic forms that recur again and again during metazoan evolution. So if we look at the different types of organisms in the metazoan family, the metazoan superphylum, there are points along the way during evolution where different aspects emerged. Multicellularity emerged when you had classical cadherins. You got tissues, separation between tissue layers, different symmetries, 3 tissue layers, and so on, segmentation. And you wound up with forms that look very different from each other and are very different from each other, but also have a family resemblance because they all express these different motifs of the metazoan toolkit. So if we look at the, from the point of view of the dynamical patterning modules, they kind of entered the evolution of animals at different stages along the way. And you had ancestral proto-metazoan cell clusters, then you had placozoans and sponges, the morphologically simplest forms, and they had, relative to the more kind of elaborate forms, they had a fairly impoverished toolkit. They didn't have everything, but as you move on during evolution, you acquire more and more of these dynamical patterning modules, and they organize these liquid materials in increasingly different ways until you get the kind of triploblastic eumetazoan organisms. Sorry, yes, eumetazoan organisms. So just to kind of recapitulate and give the very simplest example, the most fundamental dynamical patterning module is simply adhesion. By mobilizing mesoscale physical effects, ancient molecules took on new functions in the multicellular context. So you go from single cells to multicells by mobilizing the force of adhesion, utilizing a novel gene product that was partly present in ancestral organisms, but took on, had new components. So the ability of cadherins to link up to the cytoskeleton and allow cells to remain stuck to each other while they're constantly moving in relation to each other. That was something new that came in. And then you got the liquid tissue state. And the inverse, which you see in certain stages of embryonic development, is epithelial mesenchymal transformation. It's the opposite of the aggregation. So that's the simplest of all the dynamical patterning modules. So let's look at some other examples. So here's just a physical effect. These are two drops of immiscible liquid that one is engulfing the other because of surface tension. And you have similar effects when you take embryonic tissues and confront them with each other, and you get phase separation. And this actually goes on during embryogenesis, as Malcolm Steinberg was the first to emphasize. If you have a mixture of cells, the cells are different, they can sort out, they can coalesce, and you get an equilibrium situation where one layer engulfs the other. And it's a very reproducible thing because of the physical effects of the differential adhesion. And if you take fragments of the same tissue and fuse them, you'll wind up with the same equilibrium structure. So basically, there's a goal directedness here. And the goal directedness is dictated by the thermodynamics, the physics of fluid interactions. And experimentally, you can see that tensile forces govern germ layer organization. In zebrafish, you have the very same effects going on during embryogenesis. So, so this is really, physics is driving morphogenesis in a very reproducible way. And if the cells are polarized so that they're less adhesive in some regions than in one region than in others, you can get internal cavities, which we see in complex metazoan bodies and organs. And the physical analog is the formation of micelles in polarized molecules. Here, the cells are polarized, but you can get interior spaces. And this also goes on during embryo, sorry. During embryogenesis, you have self-organizing properties of mouse cells, and you have cavities forming simply by this cell polarity effect. The cells self-organize because they're polarized, and one region is less adhesive than other regions. So I won't go into the details of these examples, but the examples exist. So here is a very schematic view of the different kinds of physical effects and how they can kind of combine with each other and form different forms. And this was an early paper when these ideas were being generated. And you can see I have the Austro-Hungarian Empire behind me here. My primary evolution colleague and collaborator is Bert Miller, who's here, and Gabor Forgacs was my early physics colleague and collaborator. And we developed these ideas of how these different physical effects can generate different physical forms. And on this figure, you can see that early on, you don't need a very complex genetic toolkit to get you these effects because the environment is also contributing to kind of conditioning the physical influence on the materials. So you can have interconvertible forms from the same genetic toolkit, because it's not just the genes that are determining things, but it's the genes plus the physics. And over time, you get a kind of a drift of the developmental underlying genetic components. And eventually, you might get genomes that are just giving you one kind of form, and the forms are no longer interchangeable. So over time, you might conserve the forms, but you can change the genetics that are involved in producing, along with the physics, the forms. Okay, what about function? So organisms aren't just forms, but they're forms, they're functional forms. So the philosophers have talked about different kinds of functions, or different approaches to understanding functions. Are functions something that have survived because they've contributed to the organism's fitness, and there's a historical component, so functions are selected effects as necessary consequences of an organism's evolutionary history. Another approach is functions as causal roles. The organism is a kind of machine, and in order to keep the machine going, you have certain required functions. And so functions are just as the carburetor of an automobile is a function that is necessary to keep it going. I'm going to kind of mix these up a bit and talk about functions as appropriations of life-sustaining single-cell activity. So what I'm saying is that the functions aren't necessarily newly acquired over evolution, but they're already there in single-celled ancestors, and they're appropriated by cell types and organ types when organisms evolve. So not all animals have the same number of different kinds of cell types. All of them stay alive, all of them consume food, all of them behave in certain ways, but they don't all have dedicated cell types and organ types for performing all these functions. Some of them have many fewer, just a dozen or so cell types. We have more than 200 cell types. So this is partly optional in the evolution of animals. So here's one view of how cell types are generated. This is due to Eric Davidson and his colleagues, and this is like a complex switchboard. And you have cell types that are doing different things, and they've evolved. You have gene regulatory networks that are involved in producing the features of different cell types. And there must have been a very kind of elaborate evolutionary trajectory that gave you all these functions, but that's not really explained in this paradigm, how you got the functions, only that these functions exist and they depend on gene regulatory networks. There's a different view of cell types, which is a dynamical view, which talks about a kind of a complex landscape where you have different attractors in the dynamical system. The dynamical system can be characterized by a set of equations or a set of what's called Boolean networks, and you switch between different states of the genome, and you get different cell types that way. So again, the cell types appear sort of automatically in this case, but the genome has to be very special. It's very specially evolved to give you functional relationships among these cell types. Well, both of those models the switchboard model and the dynamical systems model depend on an idea of gene regulation that was kind of inherited from our understanding of prokaryotic or bacterial regulation. You have an active state and you can repress it. Basically the cell is like the gene is switched on and switched off. But we know now that in the case of eukaryotic cells with nuclei, and particularly metazoan cells and holozoan cells, you have a very different kind of gene regulation. You have transcription factors, but you have enhancers that come from distances along the chromosome and are recruited to the start sites. And this is something that's very different, has very different consequences. So for example, here's a current understanding. You have these domains, these compartments, and you have what are called topologically associated domains or TADs. Genes are coming together from very different parts of different chromosomes, and they congregate in these blobs. These are like liquid-like blobs inside the cell nucleus. The regulation is occurring here. And then it's you have these domains of the chromosomes. Some of them are more active, some of them are less active. Many things are going on, and it's not something that is very easy to model in terms of differential equations or Boolean networks, because what happens is that you have sometimes thousands of enhancers coming together into one of these blobs and coordinately regulating many different genes that are involved in a function. And the genes are expressed together, and the enhancers are multiple, and the strength of the expression depends on how many enhancers you have. And the genes are not related to each other by spatial contiguity as they would be in a bacterial operon, but they're taken from all over the place in the genome. And then you have these, when you have cell differentiation occurring, you have the movement of these topologically associated domains from one of these blobs, which is more active, to other blobs, which are less active, and vice versa. And when you have a situation like this, gene-gene interactions are not reversible. They're not subject to mass action. They're not stoichiometric, and they're not even topologically conserved, which means that the network structure that's so important in those older models of gene regulation, the actual connections, because of the what's called intrinsic disorder of the transcription factors, you don't even have the same network connections conserved throughout the episodes of gene expression. You have links broken and links created. So you can't say that the panoply of the cell types is a set of attractors of a genome level network or dynamical system. The transitions between cell types adjacent to one another in a developmental lineage can be modeled this way, but not the full range of cell types. So where does the range of cell types come from? And this is just what I've just gone over very quickly is described in much more detail in this paper, and for anybody who's interested. But if we look at single-celled organisms that are related to metazoans, and this is a case of a pisthecon, yeast, spatial localization of co-regulated genes is not related to genomic cell clustering as much as it's related to function. So these ancestral cells, they were doing certain things, and they had functions that were brought into effect by very distant connections between genes that were congregating together at particular sites in the cell nucleus. And I'll give one example of what I'm talking about. So we've seen this picture before, and we've seen that the epithelialization is the most fundamental cell function, this aggregation function. And it turns out that there is a transcription factor called grainy head. And grainy head is involved in all of these epithelial differentiation functions. And it's actually present in Neurospora, which is a fungus. And it also regulates cell adhesion. So basically, it's a transcription factor that is involved in a fundamental function, and it is something called a pioneer transcription factor. It opens up chromatin to wide range expression, and it's also a lineage determining transcription factor. And it's probably one of the very few that have this function. And this transcription factor pre-existed metazoans. It was involved in cell aggregation in very ancient cells, and it's mobilized in present day metazoan organisms to perform this basic function. And we can see that there are other ancestral cell functions: motility, electrical excitability, neurosecretion, detoxification, the formation of structural matrices, light responsivity, extracellular digestion, that are all related to functions that we find in animal cell types and organ types. But they were present in ancestral cells, and the genes that were involved in these pathways were regulated as a block in coordination with each other in the ancient cells. So what it seems has happened is that as animals evolved, the particular kind of gene expression apparatus with these kind of expression hubs that bring in genes via enhancers from great distances are mobilizing these ancient functions that pre-existed in single-celled organisms and turn them into cell types and organ types in more complex multicellular organisms. So let's just recapitulate. The origination and evolution of form was based on inherent morphological motifs of multicellular matter. Genetic realization of morphology is transformed over time by gene regulatory network rewiring, developmental system drift. Forms are not solutions to fitness challenges, but are inherent. They're inherent to the materials. What about the origin of cell type and tissue functions? Based on inherent life-sustaining functions of ancestral holozoan cells, the generation of novel cell types by the metazoan-specific gene expression hub system containing pioneer and lineage determining transcription factors. Functional traits are optional. They're also not solutions to fitness challenges, but they were present in ancestral cells, and they're simply recruited in a very ready fashion, not by random search so much as by simply appropriation of these pre-existing functions into specific cell types. Okay, I have just very few minutes left, and I'm going to talk about the alignment of cell agency. So here's some recent work that I did with Vijay Nanjandaya, who's I think present here, and Mariana Benitez, and student of hers. And we were looking at multicellularity in myxobacteria, a prokaryote, and multicellularity in Dictyostelium discoideum and other cellular slime molds. And you have a very great similarity in the life cycles of these organisms. And similarity is not based on any gene commonality. The genes are playing analogous effects here, and there's a certain physical similarity because the same kinds of adhesive physics and phase separation physics is used in both cases. But in addition to the generic or kind of expected physics of these aggregates and the recruitment of pre-existing cell functions, there's also one extra ingredient, which is the agency of cells. In both cases, in the bacteria and in the amoeba, the cells have individual agency. The cells are kind of searching around, looking for food, putting out signals to attract things that they consume, and so on. And when they enter into these multicellular forms through these physical effects, their agency, the agency of the cells, is kind of aligned and utilized in the subsequent development. So this is a kind of a missing ingredient of the purely physical picture and the functional appropriation picture. There is the agency of the individual cells, which is aligned and appropriated into the multicellular developmental programs. And here's a paper that we wrote that describes the essentiality of the agency in modeling these systems. We can't model them purely on the basis of middle scale physics. So to summarize, so body plans and organ forms derive from inherent physical properties of multicellular materials. New ones were added with the mobilization of new forces by new genes based on inherent life-sustaining functions. Cell and tissue functions are based on pre-existing life-sustaining activities of ancestral cells. Those functions were readily recruited to specialized differentiated cell types by an enhancer-dependent gene regulatory apparatus specific to metazoans. The agency of ancestral cells, which is dependent on their unicellular functions and activities, contributed to the evolution of development. It was domesticated along with the integration of multicellular entities. It later reemerged in part in neural crest and cancer. So basically, in a multicellular organism, you see the reemergence of cellular agency during development and in pathology. The evolutionary origin of this cell agency is enigmatic. Given the described inherencies of form, function, and agency, evolutionary change was both innovative and predictable. The role of natural selection was correspondingly limited, restricted to culling of organismal types produced by the above. Evolved organisms, in turn, resist selection by actively seeking affordances and inventing niches and ways of life appropriate to their attributes. So basically, the conclusion of all of this is that there's a great predictability in evolution. Evolution is innovative, but it's also predictable. And the agency of multicellular organisms is, to a great extent, just an expression of the aligned agency of its individual cells. I'll stop there.
Thank you very much, Stuart. I would invite everybody to unmute yourself.